So today I'll be talking on electrophoresis technique. So electrophoresis technique is one of the important tool of the clinical chemistry laboratory where a lot of biological samples like blood, urine, cerebrospinal fluid, etc. are analyzed. So introduction to the electrophoresis. Electrophoresis is a laboratory technique used for separation of charged molecule. So various charged molecule like DNA, RNA, proteins are present in the living beings. So based on their size and charge, the separation is being performed. So commonly used molecular biology, biochemistry and clinical laboratory technique. And what is its principle? The charged molecule move towards the opposite electrode when the electrical field is being applied. So it is basically based on the movement of the charged molecule towards the opposite side electrode, whether it is anode or cathode. So cathode means cation will move towards the cathode and anion will move towards the anode because anode is a positively charged while the cathode is a positively charged electrode. So this is the principle. The molecules with negative charge migrate towards the anode and the molecule with positively charged, a positive charge migrate towards the cathode. Then separation depends on the following characteristics of the molecules, like charge of the molecules, size and shape of the molecules, strength of the electric field, and properties of the supporting media. Now, types of electrophoresis. Electrophoresis can be of different types. It can be agarose gel electrophoresis. It is basically used for separation of DNA and RNA. Then we have a polyacrylamide gel electrophoresis, that is page electrophoresis for protein separation. Then SDS, sodium dodecyl lauryl sulfate, page electrophoresis, which is used for seeing the status of denaturation. The native page that is used for denaturating proteins. Then third is the capillary electrophoresis, which has got the high resolution capacity and used in DNA sequencing also. Then isoelectric focusing, that is used for separation of proteins based on the isoelectric point. Then next is the pulse field gel electrophoresis, that is PFGE, for very large DNA molecules. Now, what are the steps of gel electrophoresis? Because gel is the most commonly used electrophoresis technique in the biochemistry lab, in the diagnostic laboratories. So we will see into this. So what are the steps of gel electrophoresis? This first is the preparation of gel. So gel means on some glass plate there is a spreading of these gels, agarose or polyacrylamide gel. And second is the loading of the sample with a loading dye. And then third is applying the electrical current. The fourth is separation of molecules based on size and charge. And fifth is the visualization using the staining method like ethidium bromide, Comasi blue, and silver, or the silver stain, depending upon the type of biomolecules being separated. So we will see here, this is the pictographic of the electrophoretic technique. So this is the tub, the plastic tray in which the buffer is filled, and over this plastic tray there is immersion of agarose gel plate. The plate which is being synthesized, the film is being synthesized onto the glass plate. So agar gel is spread onto the glass plate. So this plate is used for separation. So we will fix here through some clamps. Then these are the small wells being created by using a specialized combed plate which cuts at specific ends of these agarose plate, which leading to the formation of small and micro-sized wells in which we will upload the substance to be separated. Then, and these are the electrodes. This is anode, and this is cathode, and this is anode. So, and this is the power supply through which the power is supplied. So, whole system is influenced by electricity. So, in step one, what we'll do? There will be, this is the DNA sample in gel loading buffer. So, sample is to be, which is to be loaded is prepared in the buffer. This is the loading buffer. So this sample now, this is the injector. Through this injector in these small well-like pores, the injection of sample is being given. So this is how loading of sample is being done onto the agarose gel plate. In step two, the application of electrical field is used. So on being under the influence of electrical supply, electrical field, there starts the movement of charged particles. So based on the charge and size, there will be motility at different rates of different fragments of DNA. And here at the end, at the last side, there is a well in which the marker DNA is being uploaded. So as to it has its specific pattern. So through these marker DNAs, we will be able to identify that from these different wells, the DNA samples, unknown DNA samples which are being uploaded, they contain the specific marker DNA or not. So this is how there will be a tracing by match with those of marker DNAs, right? So in step 3, what will happen? The DNA separation is, here you can see this is the agarose gel plate, and after being developed by using some staining method, so by using some stains, we can visualize these bands, different types of separated bands, which if we cannot view by simple eyes, we can use the UV trans illuminators. So through these, under the influence of UV light, these are visible, and we can see these are the marker DNA, and these are the different fractions of DNA samples, and which fraction contains which particular kind of DNA, because the RF value will remains the same of a specific DNA. So by matching with the marker DNA, we can identify the type of DNA fragment. So like in this sample, this is the DNA which matches with this marker DNA, and this fragment matches with this marker DNA. So this is how we can do the separation and identification of different DNA samples. Understood. So this is about the how the processing of electrophoresis takes place. Gel electrophoresis. Had it been there paper electrophoresis, so instead of gel this plate, we are using the paper. And paper electrophoresis has many disadvantages, as it is it takes a very longer time, because the resolution is not very as good as in case of gel electrophoresis. Thirdly, for longer time, no dipping into the buffer will lead to damage of the paper also. We will use Whatman filter paper. So in all ways, the gel electrophoresis is more good over the paper electrophoresis. So both these are zone. These are the example of zone electrophoresis. Then what are the applications of electrophoresis? So electrophoresis, as I have shown in the previous slide, that how we can identify the marker DNA. identify the DNA based on the by matching with the marker DNA. So it has a lot of advantage or use in the DNA fingerprinting, then RNA and DNA analysis, then protein analysis and purity checking is being performed, then clinical diagnostics. In clinical diagnostic for identification, for separation of different types of hemoglobin can be done. Then in forensic science, matching of different proteins. In cases of paternity disputes, we can use this electrophoresis technique. Then genetic engineering and biotechnology. Now, what are the limitations of this technique? This requires a specialized equipment. Some methods are time consuming. Then stain may be hazardous, like ethidium bromide is highly hazardous to the health. Then what are the advantages? High resolution and sensitivity. In case of capillary electrophoresis, it has got the highest grade of resolution and sensitivity, and the amount to be separated can be used in even in nanograms quantity. Then second is it is easy to perform, then requires small sample volumes, then wide range of other applications. Thank you.